A very good morning, good afternoon, and good evening to one and all. On behalf of the organizing team, I welcome you all to the webinar on ethical perspectives in planning and conducting clinical trials for cell and gene therapies. This is the fourth and final webinar in a series of 4 webinars on ethics in clinical research being organized jointly by the National Biopharma Mission and the Department of Bioethics, National Institutes of Health, Clinical Center, United States. The webinar series is aimed at building clinical research ethics capacity in India under the broad umbrella of the Indo-US Vaccine Action Program, a bilateral collaboration between the Department of Biotechnology, Government of India, and the National Institute of Allergy and Infectious Diseases, United States National Institute of Health. Biotech Consortium India Limited, BCIL, is assisting the National Biopharma Mission in organizing these webinars. We are pleased to have with us a distinguished panel of experts, which include policymakers, ethicists, and researchers, and a huge audience who are joining us from different parts of the globe through the webinar platform and YouTube live streaming. To start with the proceedings, it is a privilege to introduce the chair for the webinar, Dr. Ryder K. Lee, Professor, Department of Philosophy, University of Bergen, Norway. Dr. Ryder has a medical degree from the University of Bergen and a PhD in philosophy from the University of Minnesota. Prior to his present role, he was the director of the Center for Medical Ethics, University of Oslo. He has worked in the Department of Bioethics, NIH USA, where he was responsible for the unit on multinational research. His current research interests include ethical issues of international collaborative research and ethical issues in health sector reform in developing countries. He is a consultant to WHO and UNAIDS on international research ethics. May I request Dr. Ryder to kindly steer the deliberations of the webinar. Over to you, Dr. Ryder.
Thank you, Sanchita, for that introduction. It's a pleasure now to introduce as a welcome Dr. Christine Grady, who will say a few words. Dr. Grady is the chief of the Department of Bioethics at the U.S. National Institutes of Health, one of the two co-organizers of this series of webinars. Christine.
Yes, good morning, good evening, and good afternoon to everybody. I just wanted to welcome you to this webinar and to thank Dr. Shantita and Dr. Ryder for organizing this particular one. I know this, as you heard, this is the fourth in a series that we've been able to jointly offer. And I'm looking forward to the expert panelists that we have today and welcome you all. Thank you very much.
Thank you, Christine. So the theme for this workshops is ethical issues in cell-based therapies and gene therapy. These are exciting new developments that may give us a means to treat diseases that have been a challenge before. However, they also raise ethical issues, both in terms of assessing the risks appropriately of these. These are very complex interventions, highly specialized interventions, and also how to present the information to prospective participants. We have an outstanding panel that will address these questions, both from a regulatory policy point of view and from the point of view of scientists involved in designing and carrying out these trials. I will give an introduction to each one of the panelists before each presentation, rather than to go through the panelists all together. So we will start with the first presentation. That's Dr. Geeta Jotwani, who is Director General of the ICMR, Indian Council of Medical Research. She has a degree, her medical degree or a PhD degree in medical sciences from AIIMS, All India Institute of Medical Sciences. She is also trained in bioethics. She has over 30 years of experience in biomedical research. She is now involved in the management of biomedical research and policy making for upcoming biotechnologies at ICMR. particularly stem cell research, nanomedicine, gene therapy, and immunotherapy, which are the kinds of therapies that we are going to address in this workshop. Let me also say, before I give the word to Dr. Topani, I forgot to say each presenter will have 15 minutes. I will tell you when there are 2 minutes left of your presentation. It's therefore a pleasure of me now to give the word over to Dr. Topani.
Hello, and good morning, good afternoon, and good evening. And thank you, organizers, for giving me this opportunity to share this panel with the esteemed panelists and international experts. I have been, can you see my presentation?
Dr. Geeta, we can't actually. We are seeing an ASTCT.
Can you see now?
No.
No.
No? No. So I did share.
We saw a screen before, but not your presentation.
Okay. Is it? Okay. Can you see now?
Now we see your presentation. Yes. Can you?
Yeah. No, again it has disappeared.
You just put it in presentation mode, then we will see it.
Okay, now, yes. Sorry for that confusion.
Yes.
Yeah. I have been requested to speak on ICMR's perspective on development of appropriate guidelines and regulations for stem cell and cell-based therapies. As you all know, that past 2 decades have witnessed scientific developments in biomedical research, offering new generation therapeutics in regenerative medicine and curing the genetic disorders and other immunological disorders or immunotherapy. So just to share with you a few examples of these products, and just to give you examples of these products under stem cell and cell-based products, we have variety of stem cell products based on its potency, and there are products for embryonic stem cell, which has taken already a backseat, but now which is very promising is the induced pluripotent stem cell. Of course, there are allogenic mesenchymal stem cell products are there. There are many cell-based therapeutic products like exosomes, secretomes, and there are many combination products with nanotechnology and even tissue engineering and organoids, which are a new generation techniques and therapeutics are there. So in addition to that, if you come to the gene therapy, which is not new, but I should say that this past decades, there are many scientific developments to develop a robust technologies like CAR T cells and bringing new vectors as a delivery vehicle for the gene therapy, conventional gene therapy products, and of course siRNA, HIRNA, exon skipping technology, which is using chemical entities. We can very well alter the expression of proteins, and of course the Nobel Prize winning technology CRISPR-Cas9, which falls under the gene editing technology. Then coming to immunotherapy products, immunotherapy is a vast field. I have just put few examples here. The dendritic cell vaccines for cancer, then CAR cells. There are a variety of CAR cells now, CAR macrophages, CAR natural killer cells, and of course the CAR T cells, DNA vaccines, then cytotoxic T cells. Though these technologies, why we say this new generation technologies, these are not conventional, and these are denoted as new biological entities. They are also called as ATMPs. They are also known as new drugs now. So these are basically cell-based therapeutics, which are where we inject live cells or in combination with the delivery vehicle like vectors or many other combination technology like nanotechnology and so on. So it has been real challenge for the regulators and the policymakers like ICMR, NIH, and many other world over to derive or formulate guidelines and regulations for these therapeutics, and we still are facing challenges in the field. I'll be discussing quite a few in my next slides. So just to outline the existing regulatory framework in India, we have Drug and Cosmetic Act since 1940 and the rules 1945 and its amendments so far. Very recently, that is in 2019, we have now Ministry of Health and Family Welfare has notified new drugs and clinical trial rules to bring the clarity on this new generation therapeutics. In addition to this, to fill in the gap in between before getting a regulatory clarity on these on a regulatory front, ICMR was entrusted responsibility to develop guidelines. So the existing guidelines, national guidelines for stem cell research 2017, which are under regular amendments, and we are now preparing for 2020. Hopefully by end of this year, we'll come up with our new version of guidelines on the basis of the developments in the field and the concerns and issues and challenges we faced in India. For gene therapy products, we released last year national guidelines for gene therapy product development clinical trials, and all these guidelines and regulations are notified with the signatory from Department of Health Research, our regulatory body CDSCO, and Department of Biotechnology. So we work together in coordination to develop these documents. And in addition to these guidelines, we are working on developing a disease-specific guidelines for gene therapy, for disease-specific gene therapy guidelines, and process-specific guidelines as a addendum to these national guidelines for gene therapy product development clinical trials. And of course, we are working on stem cell therapy, a part one of stem cell therapy guidelines, which we are going to put it on a website tomorrow, that is the national guidelines for hematopoietic stem cell transplantation. And of course, we are working on developing guidelines for immunotherapy. So just to share with you the journey, as I said, that just to fill up the gap between the existing regulations for all these 2 decades, ICMR being a apex advisory body to Health Ministry and a biomedical technical body promoting biomedical research and technical know-how of these all new generation technologies, was entrusted to develop guidelines way back in 2001. Around 2001, in that time, there was more of a hue and cry about the embryonic stem cell research. So the document which we developed, first document, which is just this framework of embryonic for the guidance for the embryonic stem cell research, which was named as a draft guideline for stem cell research and regulation 2002. Then followed by that, we worked extensively to develop a comprehensive document on guidelines for stem cell research and therapy in 2007. Followed by that, on the basis of the developments in the field, scientific as well as other local issues and concerns, ethical issues and scientific issues, taking cognizance of that, we revised these guidelines in 2013, and then of course, in 2017, we released the National Guideline for Stem Cell Research 2017. In addition to these for stem cell research, even ICMR has been regularly updating the national ethical guidelines for biomedical and health research involving human participants, which is the latest version is 2017. This is our same group we are working on this document also. And then followed by that, I have already mentioned about these 2 guidelines which are coming up in a due course of time. So our journey was to just to fill in the gap, because to make stakeholders, all the stakeholders involved in developing or involved in the research activities in developing these technologies, to bring the clarity to the stakeholders that what are the requirements and how you should ethically and scientifically right manner conduct this research or develop these products, and even hand-holding for the stakeholders and direct them how the regulatory path has to be, and to bring all the agencies involved in the regulatory pathway to be on the same page. So this is the efforts ICMR has put for past 2 decades, and will continue to work towards bringing the clarity to the regulations. So as we know that the ethical guidelines for biomedical research is a backbone for all scientific and ethical biomedical research, and the principles which are described in this document are sacrosanct, and this is the backbone of ethical conducting the ethical research in the country, and this has been ratified by Health Ministry, and also is a part of new drugs and clinical trial rules as a reference. And these guidelines also put a reference of stem cell guidelines regularly. So coming to the, very briefly, I'll touch upon the ethical and scientific considerations. I know, of course, my other panelists will be speaking on the technology and the ethical issues related and risk and benefit in these various technologies. So I'll just brief you about what we considered basically while defining these guidelines for stem cell research. As you know that the stem cells are live drug. It is not like other pharmaceutical drugs or a chemical entity. So it's very, very challenging to put together the requirements for doing research using stem cells or developing products. And we know that the concerns behind this technology, that this has a tendency, because these cells have they can divide, a potency to divide and differentiate into various cell types, and they unconditionally divide. So the tumor formation, or the what we say is a very common factor which we have to take in consideration. Then, of course, the safety of the donors, because the cells we have to obtain it from the donors to make a stem cell products. So, and at the same time, even if it is autologous, it is very important to whatever the stem cells being used should be safe to be administered. So, and health of the donor, when somebody is donating, the health of the donor is very important. Of course, the collection of this stem cell, processing and storage, because there are a variety of sources of this stem cells. So it is important to guide them, and there has to be SOP for collection, storage, and processing of these cells. And processing involves various levels of manipulations. Some can be used directly as they are received, but some of them are expanded in culture, and these cells change their characteristic and function, change as you divide them or propagate them in a culture media, and then probably for the final goal, what you want to do and how you want to process them. So this is very important to look at the manufacturing and quality assurance of these products, and of course the release criteria. And the next challenging thing was there in due course, what we, as this technology was known to the scientific and clinical fraternity, we realized that there is a rampant use of this technology by the clinicians without knowing the repercussions of this stem cells. So very important to have an evidence-based application. So to have a scientific evidence that is very important before using or applying it for any disease condition. So, and that's why the challenge before the country and globally, I should say, there was a commercialization of the unapproved therapy using stem cells for various disorders, including severe injuries or genetic disorders or many incurable diseases. So I think this is the challenge world over to curb this unethical practice around stem cells for minimal manipulated cells or for homologous or non-homologous use for unproven conditions. Coming to gene therapy products, many of these concerns, ethical and scientific considerations are same for any kind of this new generation technology, considering manufacturing or quality control or release criteria. So as we know that gene therapy products are basically for the gene correction or enhancing or reducing the protein expression, gene expression. So the technology involves, it's a combination technology where vehicle to deliver this active genetic material. Sure. So correction of gene defects and protein expression, then appropriate selection of gene therapy vector is very important. Ensuring clinical relevant expression and correction happening when we apply these products and preventing unwanted side effects of these products. And at the same time, there is a misuse for unnatural advantages of gene editing or making designer babies or enhancing your physical activity or physical power for in a sports medicine. The challenges around stem cell research and therapy, which I said that the minimal manipulation is the very important category of cells which has been misused and there is a rampant practices world over, and we have been deliberating to control the use of these unethical practices. So we have finalized this definition with the help of different regulators, and this definition is totally in parallel with what is defined in 2017 guidelines, national guidelines for stem cell research. So what is the strength of our regulatory framework? The stem cell-derived product, gene therapeutic products, and xenografts intended to be used as a drug now has been notified as a new drugs forever. Why I have highlighted this? Because this will remain new drugs forever. And if you come to the next point, which I want to highlight, the academic trials which have been permissible for other approved drug products and a pharma drug license approved drug does not apply to all these new generation therapeutics, because these drugs will remain always new. Then coming to the guidelines, what our guidelines elaborate and define about stem cell and GTP products provides clear guidance to researchers, scientists, industry on different requirements in terms of donor selection, preclinical studies and clinical trial, how to design a clinical trial, how to design a preclinical studies, and then what are the personal expertise required a person involved in developing these technologies and those who are involved in administering this, and the infrastructure, facilities, CMC, and release criteria. It clearly defines the level of manipulation and approvals required, the regulatory or institutional approvals like ethics committee and other committees as defined in this biosafety committee or institutional committee for stem cell research. And also on the basis of the directions of these guidelines, we have established apex committees under the Department of Health Research to monitor this research activities in the country, that is National Apex Committee for Stem Cell Research and Gene Therapy Advisory Committee for Gene Therapy Product Development for hand-holding, monitoring, and guidance and policy making. The guidelines provides a comprehensive overview of the path initiated in stem cell and gene therapy trials within an enabling framework and with the stringent regulatory oversight. I just want to discuss little weaknesses what we have in our existing regulatory framework as per the new drug and clinical trial rule 2019, which does not elaborate on the what constitutes stem cell derived products and GTPs. It is leaving some ambiguity there but we are working along with our regulators to develop these definitions, and I think very soon we'll have a clarity on this. And they want, as per the guidelines very comprehensively defines these definitions. We need to harmonize these definitions with our regulators and then finalize them. And inclusion of these definitions in this new drugs and clinical trial rules and its amendments will probably remove these ambiguities. And product, process, and disease-specific guidelines, which I already said that we are in process of developing that. Identifying appropriate expertise in evaluating this drug product applications is a big challenge, and we are also trying to handhold and also helping our regulatory bodies to understand this, that what all expertise are required for the expert group, those who are evaluating these new generation technologies when they come to regulators for and the data which they submit. So we need expertise to proper expertise to evaluate this. Just to share with you the brief, the schematic diagram, which is delineating the regulatory requirement for these three different. I'm sorry, there is some formatting issue here in this figure. So for the gene therapy products, because it's a recombinant DNA technology, for preclinical studies, there is a requirement for institutional biosafety committee. And of course, after approval of that, there is a review committee for gene modification under Department of Biotechnology. Its approval for the preclinical studies is important. Followed by that, once they are having the approval of this, they can design a clinical trial with the approval of institutional committee for ethics committee. And this is the gene therapy advisory committee, which is doing the hand-holding and evaluation of gene therapy products and the clinical trials. And the recommendations of this committee will be forwarded to CDSCO for the approval of clinical trials. And all these clinical trials are mandatory to register with the our clinic trial registry of India. Similarly, for stem cell products, it is institutional level. There are two committees which needs to approve. The stem cell committee, which is an institutional committee for stem cell research.
Now 5 minutes overtime.
I'm sorry, I'll just in a few minutes. And then the institutional ethics committee, and then the regulators. For immunotherapy is simple institutional ethics committee, and followed by that. And CDSCO has initially had this committee to review all these protocols, and now they have subject expert committee to review. This is there at the part of our guidelines. One can go into to the links given here for our guidelines for gene therapy product development and clinical trial. And of course, Dr. Arka Shubhra Ghosh is going to discuss the designing, how to design the GTP trials. He'll be speaking in details. And this is the approval process for gene therapy I have already described. This is the process for GTP products development. The specific considerations which are here, that the structured multi-rare monitoring involved in this new generation technologies, and where these multiple committees to assist the regulatory approvals. And as per these guidelines, the germline or in utero gene therapy is prohibited in India. Somatic cell gene editing is permissible, but needs to be assured that there is no biodistribution to gonads. Therapeutic use of stem cell for unapproved indication is not permissible, only to be done under the realm of clinical trials with necessary approvals. The investigators involved in these clinical trials with GTP must have a domain expertise and good clinical practice training. Infrastructure has to be GMP certified by CDSCO. And major challenges, as I said, are to curb unethical practices around these new generation technologies. It is very important to take all stakeholders together to understand the pros and cons and the safety and safety issues revolving around these technologies. Thank you.
Thank you, Dr. Chapani, for this very comprehensive and useful overview of the Indian regulatory structure and all the details that we need to be aware of when we do these trials. We will not have any questions, answers, or discussion after each presentation. We will do that after we have finished all the presentations. And now we will go over various specific types of clinical trials in the next 4 presentations. So the next speaker is Dr. Matthew Shay from the U.S. National Institutes of Health.
Dr. Ryder?
Sorry? Yes?
We have questions also lined up.
Oh, sorry, the polling questions. Yes, I'm very sorry. So over to the polling questions.
As per NDCTR, stem cells and gene therapy are defined as new biological entities, investigational new drug, none of the above. The participants are requested to respond. We will show the results in a while. New drug is the right answer. Next question. Which regulatory framework and guidelines should you refer for developing CAR-T cell therapy? NGCSCR 2017, NGG TPD and CT 2019, NDCTR 2019, all of the above. You have 30 seconds to respond. So all of the above is the right answer. Now over to you, Dr. Ryder, for the next presentation.
Thank you. It's a pleasure now to introduce the next speaker, who is Dr. Matthew Shay from the U.S. National Institutes of Health. And he is there as staff clinician with a specialty in hematology. He has long years of experience in doing clinical trials for gene therapy trials and other trials for sickle cell disease, and been involved in counseling and the informed consent process of these patient groups. It's a great pleasure for me to introduce Dr. Shay. Please, over to you.
Okay. Can you guys see me and hear me okay? Okay, great. Well, it's a pleasure to give sort of my perspective on the clinical trials that we've been conducting here in Bethesda, Maryland. It's very nice to meet all of you virtually and to be part of this panel of experts. Over the next several minutes, I just want to take you through, what I do on a day-to-day basis when I talk to patients about the various risks that's associated, with the trials that we run. Just want to give you a little bit background of our institution. NIH, or the National Institutes of Health, is a federal research facility. We share some similarities with a lot of medical centers or academic institutions around the US or in the world, but we have some very distinct differences. Some of those similarities are that we have a heavy emphasis on research, both on the clinical side and as well as the laboratory with the bench side. We train, medical staff ranging from PhD to medical students and even fellows and residents. But we also have a lot of postdoctoral fellows and PhD candidates, as well as those in other health-related specialties. We have many institutes, but each institute function kind of like a department within a large medical facility. But the similarity, I think, stops there. We have many differences. One big one is that, we only have research patients. So all of our patients are on a protocol. And we don't have an emergency department to receive patients sort of walking in. So that's a big difference. We also don't bill medical insurance companies. So all of it, the research and the patient care is all funded through the studies. And because we draw patients from a lot of the rest of the U.S., even outside the U.S., I would say more than 2/3 of our patients are from outside our local area. And that comes, with its own sets of challenges and sometimes, ease. And a good proportion of our patients are non-English speaking. As Dr. Rydar mentioned, that I focus mostly on sickle cell disease, which is a chronic red cell disorder, and the treatments are similar to what Dr. Jawani had outlined earlier. And I sit right here in the sort of the medical provider team, but as you can see, we're part of sort of a large network of people that make this clinical trial available and approved. First, when the protocol needs to be developed and needs to be working with our protocol development team. And depending on the study, if it involves a new therapy or a new indication, we might have to involve our Food and Drug Administration. And in collaboration with them, once the protocol is developed, we send them to our institutional or ethics review board. With that, they go through several rounds of scientific and patient-related safety reviews. And once that's all approved, along with the consent forms, sometimes the Data Safety and Monitoring Board are involved if it's a large study or if it's a very intensive study that requires an external board to review the accrual and the safety. And sometimes we have data managers or even study monitors to help us make sure that we get the appropriate studies and the consent forms are correctly done. And we are on track. Once this is all done, the protocol and the consent forms are ready. And we have many repeat encounters to patients who are interested and to talk to them about the study. And then once we have provided them with sufficient explanation, they show us and they tell us that they want to be involved. So they sign the consent and we start scheduling study-related events and or procedures. And as part of our sort of stem cell-based treatments, one big target is we need to collect a sufficient number of stem cells. And whether the next step is involving a good manufacturing facility to manufacture those cells, or we go directly to our cell processing facility, and sometimes without working with our transfusion department. So the next few slides, I will go through sort of several scenarios that I encounter when I talk to our patients. So the first scenario is that, like in the gene therapy study, it's a long duration of protocol participation with many different procedures. And in this particular study, we add a normal copy of the beta globin to the stem cells to correct sickle cell disease. And as you can see with these next 3-4 bullets, it's a quite long process. There's an initial interest. We talked a lot with the patients, and then we bring them in for a screening visit. And after, I would say 3-4 encounters and many phone calls, they express that they want to join the study. So they sign, and then we start this process called red cell exchange. And after some months, we mobilize and collect the stem cells, and we might have to repeat this process to collect enough. And then they get a test dose of the chemotherapy so that we can prepare them for inpatient admission for chemotherapy and stem cell infusion. And then there's long period of cytopenias, and then there's a longer period of discharge in the outpatient setting. And then we monitor the outcomes of the stem cell manipulation. So from the start of their interest to all the way out here when they're back out in the outpatient setting, it could easily last between 6 to 9 months. And we want to follow them for even longer after that. So the question, that comes to my mind, and a lot of people ask me, is that, what risk do you disclose and at what level of details along the way? So what I tend to do is I tend to group the risk in parts, and then I first discuss the general risk of each part. But in those general risks, I make sure that I highlight those unique risks. Like, for example, in this gene therapy study, we use this HIV vector. So I talk about that and what that means and what that could mean for potentially their family. And I also highlight the worst case scenario, or the worst case complications like leukemia or death, and how often and under what circumstances those can occur. And then since the protocol process is quite long, I provide more details when they're about to partake in the next step of the study, and I give them a little bit more detail. So I do this combination approach so that I don't overload the patients with too much information, so that they don't feel overwhelmed. And one feature of the study is that the length of the follow-up when you write the protocol and the consent is that when it should be proportional to the patient's lifespan. So we have actually two particular studies for this gene therapy. The first one covers the first 2 years, and the second one, which is a completely separate protocol and consent form, covers another 13 years for a total of 15 years of follow-up. The next scenario I want to talk about is how do you disclose institutional risk? So this is sort of separate from the risk that the actual study provides. So I highlight two scenarios here that actually happened, that I happen to be part of both of them. We have a pharmacy department where medication it's a special part of the pharmacy where medication or investigational agents are compounded for clinical use. And we found out that one or maybe more than one safety cabinets that are used to prepare these medication products, and some of the albumin vials had microbial contamination. This actually led to the shutdown of this particular section of the pharmacy. But some of the patients had received the products that had passed through this section, and some of them received the product years ago. So it was decided that we as investigators had to inform all the patients from all the sections or different studies that used this section in the past years. We were to document the conversation that we had the patient, and then tell them how likely this might have affected them. So this is one way that this issue was dealt with. Some years later, in the cell processing section of our transfusion department, had water leak due to a plumbing issue. The walls surrounding the biosafety cabinets had some mold or microbial contamination. And that section of the wall, the place was cleaned up, the sections of the wall were replaced. But that of course took some time for that to be rectified, and that led to the partial shutdown or temporary shutdown of that section. But how we approached that was different. I think our institution required all of us, all the studies, to actually revise the consent form to include this risk of microbial contamination. And then once this issue was remedied, we had to revise the consent forms to remove it. So you see that there are institutional risks that we have actually two very different approaches to it. The third scenario is that several of our patients are non-English speaking. So switching gear a little bit to an allogeneic stem cell transplant that we have, where we use fully matched sibling donors to correct the sickle cell disease. The consent form is quite long. It's about 25 pages long, and it's written to be understood at grade 10 level. The patient and the donor are from Congo but spoke French. So what we do is that for patients whose primary language is non-English, we have a short form, which is just 2 pages long, that's translated into their language. And then basically that short form explains the study nature, that this is research, and that there are alternative treatments, and that there are some important contact information, so that if she has further questions, she has the information to do so. And then she signs the short form. And then the regular long 25-page form explained in detail through an interpreter. And then she signs that English version also in the presence of the interpreter. Whereas the donor spoke English better, and but we still had the interpreter present just in case that there was any terminology or concept that need to be clarified. And then he only signed the particular English consent. So consenting a non-English speaker sometimes requires sort of different approach. The next scenario is, what about the age of the patient? In the same allogeneic stem cell transplant, again, when the consent form is 25 pages long, how do you discuss the risk when the child is 7 or when the child is 15? So our usual approach is that we have the social worker to meet with the family and the young patient. And the social worker help us to identify if there are any barriers for them to understand or participate in the study. For example, can the child read, or can they read in the proficient level? Is there concerns for if the child wants to proceed, and so forth? Again, there's a shorter, what we call the assent form, that's a little shorter, that contains the common and some of the serious complication written, to be understood at the appropriate age level. And then we have the same long form, but we modify the wording a little bit. It's an adult for child consent form, except the you part is replaced with your child. And then we use the combination of both of these forms for the child and the parent or the legal guardian to sign. And then separately, I often will talk to the child separately, just to make sure that I get a sense, whether or not they know what's going on, why they're here, and if they want to participate. The last scenario is, how do you update, newly identified risk, as your study is up and running? For example, in the same allogeneic transplant study, we found that a couple of our patients had a rare bleeding disorder that developed 2 to 4 years after stem cell transplant. Both of them met the criteria for what we call unanticipated problem, which is a medical condition that wasn't previously known to be more frequent or severe. And this bleeding disorder sort of met both of those criteria. So we had to change the consent form to include this complication. What I did extra was that, as the patients, as I see them for follow-up, and when they fall into this post-transplant monitoring period, I update the patients as I see them to tell them that this is a rare complication and this might happen, so that they can be on the lookout for any signs or symptoms that of this particular disorder.
2 minutes.
And then when I see them, I also order extra blood tests just to screen for this. Since this is a particularly severe complication, it became one of the complications that I highlight when I talk to future potential candidates for the study. I think the second one is sort of similar. In the autologous gene therapy study, one patient developed mild dysplasia with leukemia potential. Although this was already in the consent form, we actually performed additional research to make sure that what was the cause, and we thought, and later on was confirmed to be chemotherapy related. So we had to revise the consent form too to include this complication to make sure that it's specifically in the chemotherapy section. And because this was a multi-center study, the sponsor wanted us to reconsent everyone of this change. And then in the future, whenever there was a change, even if it's a very minor change, like formatting change or increase accrual ceiling, they want us to reconsent again. So again, two similar situations, but dealt with in a little bit different way. So I'm about done here. Because the treatments, as you heard, and you will continue to hear, are getting more complicated, and the manipulation at the stem cell level are becoming more complex. And sometimes the procedures are long. I often need a lot of time to make sure that the patient understands the process and allow them to ask questions. And to disclose the risks that are intensive, but spread over many months, but in a small study, might need to be treated a little bit differently than those that are shorter duration but involve a lot of patients. And the follow-up period should be proportional to the lifespan of the patients. And then how do you, disclose the risk? Do you separate the risk that's specific to the study? And often that is contained in the consent form. But what about the risk that that's sort of attached to the facility? How do you disclose and change those, include those risks? And that might be different. And the different team members, whether it be medical providers or nurses or social workers or coordinators, they often offer complementary perspective on the participation of the patient. And the last thing is, a lot of the disclosure and risk are contained in the consent form. And you've heard me talk a lot about consent forms here. And they should, include the frequency, some generalized, and especially some of the more severe ones. And that should highlight a little bit of the benefits so that the patients can see the risk to benefit ratio. With that, I think I'll stop here.
Thank you, Dr. Shay, for this very interesting presentation on the informed consent process in these gene therapy trials. Then we have the polling questions.
Which of the following are important aspects to consider when drafting informed consent forms? Consent should be written at or below target grade level. Separate form needed for every young or older child. Should include contact information of key study personnel, or all of the above. Please respond in next 10 seconds. So 93% responded correctly, all of the above. Next question. Study is approved and started. Amendments are not necessary. All risks have been considered. True or false? We have the answers now. So 73% responded correctly, false. Dr. Ryder, to you for the next presentation.
Thank you. The next presenter is Dr. Maman Chandi. He is the director of the Tata Medical Center in Kolkata. He is a pioneer in the field of bone marrow transplantation in India. He has received several awards, the Pharma Shri Awards for his contribution to the medicine, and the BC Royal Award in the field of eminent medical person by the Medical Council of India. He is educated, his medical degree is from the CMC in Vellore. He is now also involved in the policy making for this area. He is working with the ICMR Stem Cell Task Force, and he is also the chair of the Department of Biotechnology of the Indian government Task Force on Human Genetics. It's a great pleasure for me to introduce Dr. Chandi, please.
Thank you, Dr. Lee. It's my privilege to be on this panel. I've lost my screen.
You can see it now.
You can see it now. Okay.
Yes.
I can't seem to see my screen, so there is a problem. Can you see my screen now?
Yes, we can see it. Can you try to move it? Do you see your slide at all?
Is that
We
all
see
right
your
now?
slide. We see your first slide. Yes, we see your first title slide.
Okay. Now, good day to all the attendees. My brief in the next 15 minutes is to quickly tell you something about what is involved in stem cell research. I am assuming that most of you are not conversant with the nitty-gritty of what a stem cell is. So I'm going to start with that area. And as discussed, we will not be talking about embryonal stem cells. Now, stem cells hold great promise for drug development and toxicity tests, to study gene control. And the huge interest now is in the area of regenerative medicine. Can we use stem cells to correct diseases for which hitherto there has been no effective treatment? Now, a stem cell is an immortal cell. It has the ability to divide, and when it divides, it always puts one cell back into the bank, and the other cell goes down to differentiate into different types of cells. Now, there are 4 different types of stem cells. When a sperm fuses with an ovum, you have a totipotent cell, which can become you or me. After about 4 days, the blastocyst forms, and here we have embryonal stem cells, which can give rise to all 3 layers, but not to an individual. Then in adults, we have multipotential cells, which can differentiate it into a limited lineage. Every tissue in the body has unipotential stem cells, which are capable of differentiating into only one cell type. So in adults, we have multipotential and unipotential cells. One of the big questions that has been plaguing science is whether there is plasticity in terms of stem cells. Now, what do I mean by plasticity? Can a hematopoietic stem cell become a muscle cell? Can it become a cardiomyocyte? Can it become an endothelial cell to revascularize the myocardium? This question in my mind remains unanswered, and at least as far as hematopoietic stem cells are concerned, plasticity is extremely limited. Now, this is an example of a unipotential stem cell, like the basal layer of your skin, where you have stem cells which can become only keratinocytes. Now, this is a multipotential stem cell, the hematopoietic stem cell, which is what I work with most of the time. This has self-renewal capacity, and it can differentiate into a macrophage, an osteoclast, a platelet, a red cell, a leukocyte, or a plasma cell. So if I want to cure a disease of the osteoclast, like osteopetrosis, I can use a hematopoietic stem cell for this. Now, in an allogeneic transplant, we have a recipient who has a disease for which there's no other treatment. Conditioning is the use of radiotherapy or drugs to ablate the patient's marrow and immune system. And the transplant itself is a process of collecting hematopoietic stem cells from the donor and transferring to the recipient. In an autologous transplant, we are exploiting the fact that we have chemotherapy whose dose-limiting toxicity is myelosuppression. Harvest stem cells, cryopreserve them, give the drug of interest, reconstitute the marrow with stem cells. Conditioning can use just drugs like busulfan cyclophosphamide, or it can be done with total body irradiation. This is a bone marrow harvest being done. The donor is prone. 2 people work on either side of the pelvis and suck out the marrow under general anesthesia because it's painful. This is a hematopoietic stem cell transplant where you're collecting peripheral blood hematopoietic stem cells after giving growth factor and plerixafor. Stem cell transplant has many complications. One is regimen related, and the late complication of some of the drugs and radiation we use is sterility. You need to counsel your patient about all of these issues before you take a patient into transplant. Infection, graft versus host disease, rejection, and relapse. This is a patient with grade 4 graft versus host disease, where the donor's T cells have gone to town on the patient, destroying his skin, liver, and intestine. Now, in any research, you apply the principles of consent that you apply for a regular approved treatment like an allogeneic stem cell transplant. For the donor, if it's a bone marrow harvest, it's the risk of anesthesia, which is 1 in a million or more. For peripheral stem cells, it's GCSF and Prolixafor. We think these are very safe agents. The problem of consenting children for a harvest is there. I had an anesthetist who said, how can you ask a child to donate stem cells? So I, in fact, brought her to the 3-year-old child who said, Doc, whatever you want, you can take from me for my brother. So even a 3-year-old can give consent, albeit with reduced understanding. With regard to the patient, the consent is done very, very carefully, explaining all the risks that I've outlined, because when something goes wrong, the physician has to face the patient and his family. And if you've not been truthful about every single part of the procedure, you end up being unhappy, and you have patients who then decide they'll go to court. This is the list of consent forms we have for an allogeneic and autologous stem cell transplant at our center. Now, the only randomized trial I have been involved with in the area of stem cell, hematopoietic stem cell transplantation, is where we decided to treat patients with 2 different dosing strategies to see which would produce better myeloablation and reduce the risk of rejection. If I had to do this trial again today, I would never do it the way I did it 20 years ago, because we tried to answer too many questions and the science was not right. So your hypothesis which you formulate must be clear, and you must know what questions you are trying to answer in the research. Leaving hematopoietic stem cells, I'll go on to mesenchymal stem cells, which can become bone, cartilage, muscle, stroma, tendon, or adipose tissue. You can collect mesenchymal cells from the skin, from the Wharton's jelly, from the bone marrow. You then expand them in culture, and then after many passages, you have the adequate number of mesenchymal stem cells. These stem cells are non-immunogenic. They are also immunosuppressive, and therefore they have been used to suppress immunity in conditions like graft versus host disease. And this is the therapeutic potential of mesenchymal stem cells in terms of immune suppression, tissue repair, and regeneration. Now, the big hype about 10 years ago was whether hematopoietic stem cells can be injected intravenously or into the myocardium, and these will help reverse the pathology of ischemic heart disease. I think the enthusiasm has come down, and that is because today we understand better that in you and me, normally in 10 hours, the same number of stem cells that we inject in 200 ml of marrow is actually circulating to the coronary artery. So it makes no sense at all to put in hematopoietic or mesenchymal stem cells either into the myocardium or into the coronary artery. I want to say a few words about preparing patients and their families for gene therapy. These slides are from a colleague in Vellore, Dr. Srivastava. Basically, we are planning 2 types of gene therapy. One is in hemophilia and the other is in thalassemia. And in both of these, how do you make a person who is just past 10th standard understand what is going to be done to his child or to him in terms of gene therapy. And therefore, the department in Vellore has been having sessions with patients and their families long before the trial is supposed to start, even to counsel them as to what is involved in gene therapy, what are the possible risks and benefits. And this is something that needs to be done in the patient community when starting on a research project. Now, in 2012, Cell Rewind got the Nobel Prize when John Gurdon and Shinya Yamanaka created IPS. Now, in Vellore, there is a plan of producing haplo-identical iPSC. So there are some individuals who share an exact allele of the HLA antigens on the other chromosome which they have, which means that the chance of these cells being matching with somebody else is much higher. So the process involves collecting blood, producing IPS, freezing and keeping the IPS. Now, one question when a donor agrees to give his blood to produce an IPS is, can I withdraw consent? Now, after I've spent a lot of money producing the IPS and the donor then says, I want to take out my cells from your IPS bank, will we allow that? The answer is no. So the consent form must include a complete fact that you are now donating your IPS for research, possibly for subsequent treatment. The next question arises, suppose we have a really nice IPS cell line and a bank. The donor asks, these are healthy, normal donors. Will they be paid? Suppose you use these stem cells to treat diabetes because you now have generated beta cells from these mesenchymal stem cells. Will I be paid? The answer is no. And one must be very clear about funding and the consent process when you ask a person to donate cells for a haplobanking project. One of the things I have found whenever asking for consent is whether I can truly ask myself, if I am in the patient's position, will I be willing for this research, this randomization? Because I know much more about the issues than the patient does. So if a researcher can honestly ask this question of himself and answer it, then it's likely that nothing will go wrong. So as in this concluding slide, will I be willing to be a subject if I was in the same position? The other issues in the ethical process, the consenting, the DSMB, all of these have been covered by the previous two speakers. Thank you very much.
Thank you, Dr. Chandy, for this very comprehensive but still concise overview of all the issues raised by stem cell therapies. We now have some polling questions again.
Induced pluripotential stem cells will be of use in the following conditions: for drug trials at the cellular level, for allogenic hematopoietic stem cell transplantation, for creating cell banks of HLA mismatch. Please respond in next 20 seconds. So for drug trials at cellular level is the right answer. Can we have the next question, please? For stem cell research, the guiding principles which should be followed are maximum benefit for society, patient who enters the trial must be compensated financially, DSMB is necessary. Thank you. The last option is correct. Now over to you, Dr. Ryder.
Thank you. The next speaker is Dr. Vikram Matthews. He is a professor and head of the Department of Hematology at CMC Vellore. He has been involved in the research and management of leukemia over the past 20 years. Specialty training is been in the United States at the University of St. Louis in Missouri. He has worked on various issues related to prognostication and treatment of leukemia, and specifically of novel protocol of using arsenic trioxide as a frontline therapy of acute pyomyelolytic leukemia. He is currently involved in the evaluation of leukemia with a focus on the role of microenvironment and drug resistance in leukemia. Please, Dr. Matthews.
Good evening, everybody, and thank you to the organizers for having me. Could you see my slides?
Yes.
Okay. So I'm going to talk a little bit over the next 12-15 minutes on risk communication to participants in CAR T-cell therapy. Most of the previous speakers, Dr. Matthew Shay, Dr. Maman Chandy, have gone through the philosophy of the principles of the consenting form and the consenting process, both at a cross-sectional prior to study and an ongoing process by which things have to be modified. What I'm going to do is actually talk to you a little bit very broadly on the chimeric antigen receptor construct and the principles of CAR T therapy, a little bit about CAR vector manufacture at an industrial level, kind of a GMP, which is going to be addressed by the next speaker. These two topics I will not be covering in great detail. I'll be talking to you a little bit about production of CAR T cells, the toxicity profile and the potential risk, center and expertise required in the context of risk mitigation potential, and talk about a risk-benefit analysis and decision to proceed with CAR T-cell therapy. And finally, I'll touch a little bit on current regulatory status and international and national issues. So the principle of a CAR T-cell is essentially to harness the best of what's called the humoral immune system and the cellular immune system. The humoral immune system is essentially antibodies, which has the unique feature of being very specific for a particular epitope. But in terms of efficacy, it is not as good as the cellular system. The cellular system, on the other hand, requires MHC-restricted recognition of antigens, and there could be some amount of cross-antigen. So there's less specificity, but it's much more effective, and in terms of cytolytic potential, is very high. So in a CAR T cell, what one actually does is conceptually brings the immune recognition epitopes of the antibodies, gets rid of the entire MHC restriction of CAR T cells, links it to the activation component of the T cell, and now the T cell can use the specificity of the antibody and bring about cell kill very effectively against a target of interest, which in this case, most of the times is a cancer-specific epitope, which is not present on other cells. So this gives you a sense of the construct. There's the heavy and light chain, usually linearized with a linker and a hinge molecule, a transmembrane domain, co-stimulatory molecules, and stimulatory, usually a CD3 zeta immune receptor tyrosine kinase activating motif, which brings about the activation of the T cell. And the CAR T cells, again, illustrating the combination of the benefits of an antibody with the CAR T cell, the T cell receptor. And there are basically generations of CAR T cells. Broadly, the early CAR T cells only had the activation component, which is a CD3 zeta. The subsequent generations have additional co-stimulatory molecules, either CD28 or 4-IBB. And the third generation, that's the second generation CAR T, and the third generation has often two co-stimulatory molecules. And the obvious objective of this exercise is to increase the potency and efficacy of these agents, and also the potential for these cells to live long-term within the recipient. And finally, there are additional modifications where the CAR T cells also produce cytokines, and these are T cell receptor-directed universal cytokine killing cells, which in addition to the ability to recognize epitope, also release cytokines which can bring about cell kill. Many of the elements of this, like for example, just the length of the linker, the hinge structure, can have significant effects on how the cells function, both in terms of efficacy and in toxicity. And that's not something that I'll be going to in detail because that involves a lot of vector biology. I will also not be talking about the actual vector manufacture and the way that that is produced. But once that is produced, we have to actually produce CAR T cells, and I'll talk a little bit about that. I'm going to talk mainly about anti-CD19 directed CAR T cell therapies because that's what has really gone the furthest and we have approved indications. This is a crowded slide. I'm just going to walk you through one just to give you an illustration of the variations. Tisgenlecleucel has been approved. It was initially developed in UPenn, was subsequently licensed by Novartis. It has a CD8 hinge and transmembrane domain. It has a 4 IBB co-stimulatory molecule. The starting cell population is peripheral blood mononuclear cells, and the vector is lentivirus. So similarly, there are other vector constructs which have different hinge transmembrane co-stimulatory molecules and with different targets. While we're talking about CD19, there are also CAR Ts that can direct against various solid tumor antigens and epitopes that are specific to other malignancies as well. Again, just to illustrate that some of these products are out there, 2 of them here have already been approved. One is the axicabtagene, and this is tisgenlecleucel. They've been approved for DLBCL in the case of the commercial name, which is Yescarta, and the Cambria has been approved for DLBCL and for ALL. Both these malignancies, that's the lymphoma and the leukemia, express CD19 on their surface and can be targeted by CAR T cells. And usually the inclusion criteria for patients who go for these therapies are patients who have had multiple lines of therapy, and essentially without this therapy, their chances of survival are practically nil with available therapies, which they have most probably exhausted the standard care approaches to therapy. Now, once you have a CAR T cell vector in place, how do you manufacture it? This slide is courtesy of Miltenyi Biotech. Basically, one option is to take the patient cells, ship it to a factory, which could be, say, from Chennai to Delhi, if the manufacturing center is in Delhi. In Delhi, they put the construct and the cells together in a GMP facility, produce your cells, and then ship it back to your hospital in Chennai for infusion. The other option is to have a decentralized or a production done at the center. This has numerous advantages in terms of reducing the cost, as I will illustrate later. There are standard release criteria once the CAR T cells are produced, but obviously all the caveats of safety have to be addressed to, consistency of the product, safety of the product, and reproducibility of the manufacturing process. This is just to illustrate in one of these decentralized manufacturing process. This is done at Case Western Research Unit with the Miltenyi product. This is a closed system called a Prodigy system. You can see I put this picture up just to illustrate that the machine literally sits in an ISO 7 or a class 100,000 facility in a regular clean lab, does not require any high-end GMP, and you can imagine how this significantly cuts on the cost of production. Now, this is a GMP facility that's in our institution. We have a Prodigy system. We don't, at this point, manufacture CAR T cells, but we use it for other reagents. But conceptually, it would look like this. You would get your product from the patient, the lymphocytes or peripheral blood mononuclear cells. You would put it into this closed system. It would go through a series of steps, which I'm not going to go through, but this is all done within the machine in a closed system. At the end of 12 to 14 days, you have your transduced T cells. If they fulfill the release criteria, they can be released to be used on the patient. The Department of Hematology, CMC Velo, our cell therapy program, we have a transplant program that was started by none other than Dr. Maman Chandy way back in 1986. We have a large transplant program by any standards in the world. We do almost 250 transplants a year. We have a class 10,000 facility, the Clinimax. The Clinimax right now we're using on various studies that are going on. We have a TCR alpha-beta depleting graft manipulation process. We have CD45RA depleted prophylactic DLI in the setting of haplostem cell transplant. Viral CTLs, we have standardized and initiated the CMV CTL work. The multiviral preclinical work is pending. We have an optimized NK cell trial that is going on, which is recruiting patients. And we hope to have our CAR T cell product also going within the same system. And for all the advantages, we feel that this is a strategy that we can go forward in India. Now, when we talk about risk, when I talk about CAR T cell production and center effect, it's important to remember, even in the best of hands, in the tisgenlecleucel study, for example, by Novartis, in about 10% of those patients where the product was shipped to the company, at the end of the day, they could not get a successfully transduced product. And when you have a centralized manufacturing process, there's also the requirement for cryopreservation and safely shipping these products cryopreserved. Your target viability is about 80%, but often the viability can drop in these processes of cryopreservation and shipping. And you also have to have, in the context of a decentralized manufacturing process, consistency of your manufactured process. So what is the risk here? The risk is one has to counsel the patient. The potential failure to get an appropriate product in time could have life-threatening consequences. Remember, the patients going for these therapies have exhausted usually all standards of care and are essentially depending on this product to salvage their disease. And at the end of 4 weeks of waiting, their disease is likely to progress and you will not have your cells that you plan to infuse in these patients. And that's something the patients must understand is beyond your control. When you talk about CAR T cell production and center effect, I showed you some of our data just to show you volumes that we do. The volumes are important to some extent because transplant is not about individual brilliance, it's about teamwork. It's about nurses, it's about teams who understand the complications, it's about good intensive support teams who can recognize complications and intervene early. It's important to counsel patients, and if it's the first time you're doing certain therapies, you have to counsel them that you have certain expertise and experience, but certain things are new. There are experiences that most transplant centers who do large volumes of haplotransplant will have, but they will not have the kind of complications that we see in CAR T cell therapy. So early recognition and interventions are ways of mitigating these therapies, and that is very dependent on the experience and expertise and training of the transplant team that is involved with this therapy. When you look at the overview scheme of a CAR T cell therapy, it will look like something like this. You have a patient who has got refractory disease, usually after multiple lines of therapy. You give him some therapy to get his disease under control. Then you harvest his peripheral blood lymphocytes, like Dr. Mamanchandi had told you, through on a peripheral blood apheresis machine. And then it takes about 2 to 4 weeks for it to take you through the process, whether it's centralized or decentralized, to actually manufacture your CAR T cells. But before you infuse your CAR T cells, you usually give some kind of therapy to immunosuppress the patient to accept these modified T cells, which are the patient's own cells, but they could potentially get rejected. Or you could occasionally, nowadays, people also use allogeneic cells. And the lymphodepleting protocol usually is a combination of drugs like fludarabine and cyclophosphamide, which have their own toxicity problems. Once you infuse the CAR T cells, usually the maximum complications happen in the first 4 weeks, and those are things that you look for, and you also look for long-term efficacy and persistence of these T cells and long-term toxicities. So CAR T cell therapy, Dr. Geeta Jotwani used the term unique living drug. Remember, conventional PK/PD kind of work that you do with a small molecule or a chemical agent will not work here, because these cells will interact with the host and the immune system of the host can reject or enhance the effect of the cells that you infuse, can affect their proliferation survival depending on the patient and the disease burden. So the efficacy and toxicity can be very variable with the same product. These are the kind of toxicities that you go, and I'm going to walk you through this in the order in which they are likely to appear. So when you have a patient, as I walked you through the process, first you have your lymphodepletion process. After your lymphodepletion process, the cells are infused. You can have certain complications like anaphylaxis. Then you can have what is called on-target, on-tumor toxicity, and I'll talk a little bit about that. And you can also have on-target, off-tumor toxicity. I'll talk a little bit about that. And finally, the most common toxicities that one talks about and dreads is severe cytokine release syndrome and neurotoxicity. I will not be talking about cross-reactivity because, as I told you right in the beginning, the epitopes and the antigen, the variable region that we use to recognize the epitopes are fairly specific, and cross-reactivity is usually not a problem. Similarly, insertional mutagenesis and leukemia is a theoretical concern, but in fact, the incidence is extremely uncommon. There's only one case that has really been reported, and we think that this is an extremely small risk. I will not be talking about in detail. When you talk about effects of lymphodepletion, these are usually the drugs you use before infusing the CAR T cells. Often they're a combination of fludarabine and cyclophosphamide. They have their own toxicity profiles, and you should not confuse them with the toxicity that can follow after infusing CAR T cells. So, for example, these drugs can cause cytopenia and hemolysis. Fludarabine can actually by itself induce a neurotoxicity with white matter demyelination and can mimic an immune-mediated neurotoxicity. And there's an increased risk in the setting of renal failure. There are foreign proteins that are used in the production of these CAR T cells, and rarely you can have an anaphylaxis to those particular proteins. When we talk about an on-target, on-tumor effect, imagine B cells which express CD19 and you put in your CAR T cells. And these CAR T cells go and very effectively and very rapidly destroy these cells. This massive destruction of these malignant cells can result in the cells breaking within the circulation, leading to hyperkalemia, hyperuricemia, and renal failure. What do we mean by on-target off-tumor effect? So if you have a CD19 CAR T cell, it will hit all cells that express CD19. So in addition to your malignant cells, normal B cells that are involved in producing immunoglobulin also express CD19 and are likely to be destroyed. As a result, the patient will have no ability to produce immunoglobulin and will need IV immunoglobulin replacement. This actually, in the context of CD19 CAR T cells, is a relatively benign problem because you can efficiently replace the effect. But in many solid tumors, this on-target off-tumor effect can be devastating. Cytokine release syndrome is something everybody talks about. It's essentially what happens is you have a rapid activation of CAR T cells and immune cells that are infused into the patient. As they interact with the antigen that they are supposed to interact to, they undergo activation and release a whole lot of cytokines. The clinical features can be a mild flu-like syndrome to life-threatening vasodilatory shock, capillary leak, and organ damage. Numerous cytokines are involved in this. Usually occurs within the first 14 days of CAR T cell infusion and as early as 2 days. Significant variation in incidence and intensity. The variables are not that well understood, though there's a lot of ongoing work. The grading system has been challenging. The American Society for Transplant and Cell Therapy came up with a consensus guideline for people interested. This is an important document to read. It's in the Biology of Blood and Marrow Transplantation in 2019. Essentially, it grades CRS. And early stages is just a bit of temperature. By the time you reach grade 3, you need vasopressors to maintain your blood pressure. And by the time you're grade 4, you need ventilatory support, usually some kind of mechanical support. The therapy is usually an IL-6 antagonist, tocilizumab, and steroids. Newer agents include cetuximab, anacrina, IL-1 antagonist, and also chemotherapeutic agents like cyclophosphamide and other drugs like dasatinib have been used. The other toxicity that we really worry about and talk about is neurotoxicity. Often follows a cytokine release syndrome, but not necessarily always. The terminology used is ICANS, or immune effector cell associated neurotoxicity. Again, it's well defined in the document that I talked to you about, usually preceded by a cytokine release syndrome. The increased CRS, both the intensity of the CRS, the greater the risk of ICANS. Early findings are often very subtle, just word aphasia, inability to remember names, etc., which can gradually progress to global aphasia, and its terminal stages can lead to coma and fatal cerebral edema. The variables, again, are not well understood, though there's a lot of theories, but there's an increasing attention to endothelial cell damage and to the use of anacrina, which is an anti-IL-1 antibody, which seems to work better in neurotoxicity, while tocilizumab does not seem to work in the context of neurotoxicity. But the management is essentially centered on early recognition and early use of steroids. There's a grading system, which is there again in the STCT guidelines, which involves daily assessment of what's called an encephalopathy score, along with features of decreased levels of consciousness, seizure, motor findings, and features of elevated cerebral edema. So in terms of incidence, this is a crowded slide, just to give you a sense of CRS grades. What can you expect in some of these trials? It can vary from 93% to 37%, but severe CRS, the cytokine release syndrome, is about 13% to 22%. Neurotoxicity, all grades can vary from 65% to 21%, but the severe types that can vary from 31% to 12%. The newer constructs and clinical trials show lower incidence of these terrible complications. Current regulatory status, these are the agents that are currently approved: Testen-Gleclusil from Kymriah, Axicabtagin, and the new one called Brexacabtagin is the first FDA-approved CAR T-cell therapy for mantle cell lymphoma. There are numerous ongoing trials, majority of them in USA and China, more than 400 ongoing clinical trials, 171 in the US, 152 in China as of February 2018. So I would summarize the risk and communication as one needs to talk to the patient about the risk benefit, and who are the patients that are usually taken at this stage for these kind of interventions are patients who have exhausted practically all standard lines of therapy. One must talk about the risk of failure to get an adequate product, must talk about the center expertise and experience in handling complications, toxicity of lymphodepleting therapy, the rare risk of anaphylaxis. One must discuss on-target, on-tumor effects and on-target off-tumor effects, the significant risk of cytokine release syndrome and ICANS is something that has to be discussed with the patient, including life-threatening progression of these complications and the rare risk of insertional mutagenesis. And finally, don't forget, even with this therapy, the majority of patients relapse, and patients must be clearly told, while this therapy is probably the most effective in the state that they are, it still does not guarantee a cure. I think I'll stop there. Thank you for your attention.
Thank you very much for this overview of CAR T treatments, and in particular the focus or the review of the risks involved in these therapies. Then we also have some polling questions.
So the first question, a common adverse event with CAR T cell therapy is myocardial infarction, pulmonary embolism, cytokine release syndrome, insertional mutagenesis. Please select your option. So we see the audience response now. Cytokine release syndrome is the correct answer. We have the second question now. Immune effector cell-associated neurotoxicity syndrome, or ICANS, can be present with peripheral neuropathy, word aphasia that may progress to global aphasia, subdural hemorrhage, viral encephalitis. So we see the response here. The second option is the correct option. Over to you, Dr. Ryder, for the last presentation.
Thank you. The next and the last speaker is Dr. Akasubra Ghosh, who is director of the GROW Research Laboratory, Narayana Netrayaya Foundation in Bangalore. He has his PhD degree in molecular medicine from the University of Missouri in the United States. His current research interest focuses on understanding of the genetic and molecular signaling mechanisms driving pathogenesis of ocular disorders and cancers. The GROW laboratory, where he is a director, is developing a database of mutations and genes associated with genetic diseases within the Indian population for future recombinant vector-based gene therapy and cell replacement modality. And a clinical-grade gene therapy vector production platform is being developed, and various studies are being done with the aim of performing human clinical gene therapy trials in India. Dr. Ghosh, over to you.
Thank you. Thank you, and good afternoon and good morning, everybody. So today what I'll do is, since a lot of the ethical considerations in general across regenerative medicine have been discussed, I am going to talk about the technical aspects that we need to take into consideration as we try and develop gene therapy vectors for human trials. So I hope everybody can see my screen. All right, so let's first start with defining what are gene therapy products. And this definition that I'm mentioning here is now part of the National Gene Therapy Guidelines that Dr. Jotwani had earlier referred to. So essentially, gene therapy products, or GTPs, are in any form the extra genomic DNA or RNA segments that are used for any kind of treatment. So these extra genomic DNA or RNA segments have to be delivered into the tissues, the organs, or the cells of a patient. So that delivery system typically is derived from viruses of different kinds. So these are recombinant viral vectors A lot of these belong to the adenoviral, the retro or lentiviral, and the adeno-associated viral vector subgroups. We also have non-viral vectors, and these would be chemicals or nanoparticles or liposomes, etc. And we'll talk a little bit more about them in the following slides. What comes next are oncolytic viruses. These are viruses which would always kill the cell that it infects, and these are being tried out for treatment of various kinds of cancers. The new modality for doing gene therapy currently is, of course, gene editing, and the gene edited products might also be considered as a GTP. And in the following slide, I'll explain how regulating RNA using short hairpin or small interfering RNA molecules are also considered the GTP, and there's some upcoming new vectors being developed from bacterial or microbial sources, which can carry even a chromosome. So essentially, gene therapy can be split into two kinds. One is in vivo gene therapy, where you directly provide the DNA element or RNA element by a vector, which can be a virus or a liposome or whatever, into the body. This can be directly to the local tissue that is affected, say the eye or the muscle or liver, or you can have it intravenously given. And the other one is ex vivo gene therapy, where you take cells from the patient, and then outside the patient's body, you manipulate it to fix, say, a broken gene, or you introduce new synthetic genes like you do in case of CAR T-cell therapies or stem cell therapies, and then you put it back to the patient's body. So both of them would come under the umbrella of gene therapy. So to begin with understanding the risks associated with doing gene therapy, we first have to understand the vectors and what are the requirements of a gene therapy vector, and what are the advantages and drawbacks of those. So broadly, we can separate them into viral and non-viral, and I've listed these here. But what is important to understand is that an ideal vector has to have the carrying capacity for the gene or the DNA element that you want to put into the patient. It should preferably be undetectable by the host, by the patient's immune system. It should not be toxic. It should not be inflammatory. And we must remember that in almost all these diseases that we are trying to treat, there are pre-existing conditions which cause inflammation and other things in these subjects. So the vector, the treatment that we are trying to give them, that should not exacerbate that condition. Finally, the goal of gene therapy always is to have a long-term effect of the treatment, so that we don't have to come back with weekly or monthly injections. So with that in mind, the broad advantage and disadvantage of these is listed here. So viral vectors naturally have very high capacity or efficiency of delivery of the gene, whereas non-viral vectors do not have that. The disadvantage of viral vectors is that they typically are associated with high immunogenicity to the to viral vector derived proteins. It could be the capsid protein or remnants of its genome. The other problem with viral vectors is the production process is very complicated and technologically challenging, and thus extremely expensive. Whereas in non-viral vectors, the production process is easier, and therefore it is less expensive, and it has a better safety profile. But because of the significantly lower delivery capacity of the DNA element, most of the current trials do prefer viral vector backbones to deliver. So if you look at the gene therapy vector trials, you have a variety of these viruses, adenoviruses, retroviruses, which constitute a large number of the trials ongoing. This is a little older data. Nonetheless, what's important to understand, if you look at the list, there's only about 20% of the trials which include a non-viral modality for gene delivery. So for the next part of my talk, I will be speaking more and more about the viral vectors. And in case of the viral vectors, it's very important to understand which kind of viral vector would be useful for the kind of disease that you're trying to target. So adenoviruses and AAVs, these are, and HSVs also, an important consideration for them is that they can infect both dividing and non-dividing cells, and they are non-integrating. There is a low level of integration that has been reported, but still that is part of the safety consideration, that they don't integrate into the host genome, whereas the retro or lentiviruses have to integrate into the host genome. So that can cause insertional mutagenesis. The second consideration is the size of your gene or the entire regulatory element, regulatory cassette that you are trying to deliver. And that's where packaging capacity of these vectors becomes important, where AAV has an important limitation that it is very small. It can only take, it cannot take the larger genes. So we have to bear in mind which can be used for what kind of diseases. Now, the other more pressing consideration, of course, is how long the gene that you transduced is going to be expressed in the patient cells. So there, AAV and as well as the lentiviruses, they have the advantage of having long-lasting expression, whereas adenoviruses and vaccinia viruses have very transient expression. So they would be more useful for hit-and-run kind of therapies. So these are just examples of the kind of trials, the kind of diseases that are being treated with AAV. You can see there are monogenic diseases like neuromuscular diseases and ocular diseases, as well as multifactorial diseases like AMD and Parkinson's. And similarly for lentiviruses, there's a major proportion of them are in cancers, which would include the ex vivo gene therapies like the CAR-T, CAR-NK, etc. So this is a slide, this is a image that Dr. Jotwani had earlier shown. So this is part of the guidelines. And what it essentially shows is that the entire gene therapy product development can be broken down into these four primary segments, if you will. First is the GTP development, then the GTP production, then the preclinical testing, and the clinical trial design. And all of the risks associated with the administration of a GTP in the patient, a lot of that consideration has to go on during this process of development, production, and testing. Because together we can, by keeping these specific points in mind, we should be able to reduce the risk to the patient significantly. So during GTP development, we must very carefully understand the vector, the components of the vector, particularly the transgene design, the regulatory elements, so that you do not have off-target effects, etc. And also determine through the preclinical testing which route of administration is ideal, so that you can determine the effective doses required. So for certain conditions, you might have to go the intravenous route. Certain conditions, you can do local injections, and the dosages required will be different for those. The second thing to be considered, of course, is the biodistribution and the immune cell activation or immune activation that is specific not just to the vector, but also to the transgene. So these are the things that we need to know about the GTP itself before moving into the clinical trials. And then in the clinical trial design, of course, you have to have the patient details, genetic background, and a lot of these factors that I'm going to talk about in the following slides. So this slide here shows a typical gene therapy vector production process. So it's a long process where you use these the production cells in which the viral vectors are going to be made. Like I pointed out earlier, in case of nanoparticles or in case of chemical or liposomes that are used for delivering the DNA, this process is not at all that complicated. It is chemically synthesized and typically is a few steps only. But in case of viral vectors, you have to produce it in a producer cell line like 293 cells, etc. And then it has to be produced to a scale where at the end, when you have the viral vector, that is at a dosage that can be administered for appropriate clinical response. So during this process, there's a lot of chances of having host cell proteins, host cell DNAs or RNAs that can contaminate your final product. And these are very important considerations which have to be removed through repeated multi-step purification processes, multi-step polishing processes, maybe multiple chromatographic graphic processes, etc., to reach the final product which is safe to administer. And the knowledge of this is very important. So essentially, if you have to broadly categorize the risks associated with these vectors in clinical trials, it can be divided into two: the vector-associated problems and the trial-associated problems. So the possible effects that can happen because of these are: A is in the vector design itself, where you have unrequired DNA elements or RNA elements that are part of your expression cassette. So those should not be there. And second is in your production method itself. So ideally, you would want the transgene itself to be synthesized to be less immunogenic. So the host proteins, etc., must be removed. In the clinical trial part of it, you have to consider the age and severity of the disease. You have to consider comorbidities, and you have to consider other treatments ongoing to find the correct route of administration. So here again, we delineate the essential risk sources from vector design and production methods, which can come from the vector envelope, the genomic sequences, and the encapsulation of the vector if you're using an encapsulation agent, residual contaminants, and we also have to consider yields. So all of these have possible solutions and possible steps at which these can be intervened in to correct. An example of a possible problem is in these muscle disease trials, where a high amount of vector was given to certain patients, which resulted in severe adverse events. So that is a challenge that we face, particularly in diseases that are body-wide. So in order to mitigate such risks, we must also consider the risks from the clinical trial design. This comes from patient inclusion-exclusion criteria. So you must know the type of mutation, presence of other mutations, comorbidities. We must use the best possible clinical endpoints and not miss those. And of course, status of the immune response, which comes from the neutralizing antibody titers, the T-cell mediated response, and hepatobiliary responses, which essentially boils down to inflammation and organ damage. So in order to do these, mitigate these, the best way is to do detailed preclinical trials, and for across all the organs of the animal model that is used. So finally, some considerations for a gene therapy trial are you have to be very critically examine what your inclusion-exclusion criteria are. There has to be detailed disease profiling. The natural history of the disease in the patient that you're enrolling must be well defined. Of course, mutation and inheritance patterns and detailed prior treatment history. So in order to communicate these risks to patients and the patient caregivers, you must inform them that these gene therapy trials are experimental and carry significant and a lot of times unknown risks, because the rodent models or lower mammal models don't always replicate all the immune responses that we see in a patient. And gene therapy trials are very long. They are multi-year. The patient, therefore, has to agree to diligently do the follow-ups, there may be biopsies and things required, which are invasive tests. And a significant amount of counseling needs to be done by the treating teams. Of course, a lot of testing, blood tests need to be done before you inject. That includes immune cell parameters. And finally, it is important to focus on holistic wellness as well. So therefore, quality of life questionnaires and quality of life data from the specific disease group is very important in trial design as well. So with that, I am done. So for gene therapy trials, there is a lot of hope, but both patients and treating teams must prepare for setbacks.
Thank you, Dr. Ghosh, for this very good overview of all the things one needs to consider in these gene therapy trials. We now have some polling questions.
Which GTP production hurdle is most important for ensuring patient safety? Vector yield, immune activation, carryover contaminants, all of the above. You have 15 more seconds. All the above is the right answer. The next question. Which trial selection criterion is essential? Genetic background, accurate diagnosis, hospital setup, patient consent. Here we have the audience response. Patient consent is the most essential. Dr. Ryder, over to you for discussions.
Thank you. Thank you very much. And thank you to all the speakers. This has been a fascinating overview of all the things that goes on also in India and what the possibilities are for research in this. And I think we have just scratched the surface. We have raised a number of ethical issues that need to be attended to, how to communicate risks, for example, what kind of information to provide. I don't think we have time for a discussion at this moment. I think we would have to leave that for another seminar where we focus more on the details of the information process and so on. What we have left now is some concluding remarks from the two main persons who have been instrumental in getting this workshop, series of workshops going, Dr. Kavita Singh and Dr. Christine Grady, who will say a few words. But before we do that, I will ask Dilip if there's anything else that we need to do before we make the concluding remarks.
I just want to mention two quick announcements. One is that I request the attendees to fill the feedback form for this webinar. And the video of the webinar proceedings will be available on the NBM Workshops website. And you can continue with the concluding remarks, please. Thank you.
Okay. Anything else, Dilip?
No, Doctor.
Okay. Then I give the word to Dr. Kavita Singh.
Good evening, everyone. It's such a privilege and honor to speak after all of you. And honestly, nothing I say would be able to beat the scientific knowledge all of you have so transparently, honestly, and committingly have shared in the last 1 hour. And also the seminars or the webinars, we've had a series of webinars in the last 6 weeks or now. And Dr. Christine is here. I think Dr. Christine and Dr. Ryder, we met about a year back. We're all planned actually a physical meeting. The last we planned, we were scheduling these webinars across the country at 2-3 places. The plan was that we'll have large groups, we'll have more focal discussions, we'll have small group discussions, large discussions. But at least we could have what we did even this was extremely, extremely beneficial. And that I would say is because of the participation each webinar has seen between 350 to 500 people, and especially for a topic like today's, which is such a new topic for discussion, but we are seeing 300 plus participants. It speaks for the quality of our speakers and for the agenda and for the content of the topic which was planned. So I would not take much time, but a sincere word of thanks from behalf of Department of Biotechnology, Biopharma Mission, the Department of Bioethics, the US VAP program. There's so many organizations and programs which are thankful to all our faculty members who have participated and shared their knowledge with our participants. And thanks to BCIL for so diligently following up with the participants and with the speakers. Thanks, Dilip. Thanks, Sanchita. Thanks to Dr. Purnima. And I can't thank all the 2,000 participants, but thanks to all of you. It just gives us the impetus to plan more of such interesting topics and inviting all these experts to our forums. Dr. Ryder, thank you once again for being such a wonderful nodal point and not giving up in midway. Thank you so much, everybody.
Dr. Grady, Christine.
Yes, I really just want to add my thanks first to Dr. Kavita Singh for being willing to go on this adventure with us. And as you just said, it was almost a year ago that we met the first time, and the idea was to have a series of in-person meetings. I want to thank everybody, but especially Rydar Lee and Shreya Malik, and for making sure that we could transform the idea of in-person meetings to this series of webinars. I think they have been very successful. I've been very impressed by the number of people in each of them and also the quality of the presentations, and a lot of territory has been covered. Last but certainly not least, I want to thank Dilip Joy and Dr. Sanjita Chowdhury for running the meetings, each of them very professionally and very all the technical details, etc. And I want to thank Dr. Rydar Lee for being the sort of mover and shaker on our side in terms of making sure that this actually happened. So thank you all, and I hope the rest of your day goes well.
Thank you.
Ryder, if I could just add a word, what you do, you always take your team for granted and you forget to thank them. So Mahesh and Miraj, who have been I have not done much. I really should not be taking any credit. So thanks to Mahesh and Miraj, who are the vaccine workers and have been working diligently again. Thanks to both of you.
Yeah. Now, let me just express from my side, working with, so on the day-to-day basis with all of the people involved, your staff and so on. It's been fantastic to do that. Very responsive, very professional. It's been a great pleasure to be able to do this. So I'm looking forward to doing more of it.
Yeah.
Thank you. To conclude, it's on behalf of BCL also, I would like to express sincere gratitude, particularly to Dr. Kavita Singh, Dr. Ryder Lee, Dr. Christine Grady, particularly for guiding us in conceptualization and execution of these webinars. It has been a great learning experience. I would also like to express sincere thanks to my colleagues, Shreya, Dilip, and Rajesh, for doing the needful to ensure that these become successful. And we look forward to many more such interactions in the future. Thank you so much.
Thank
We look forward to, please have safe times ahead. Good night and good luck. Thank you.
Thank you.
Thank you.
Thank you.
Goodbye.
Bye-bye.
Bye.
Bye-bye.